Chapter 2 Biological Classification. Since the dawn of civilization, there have been many attempts to classify living organisms. It was done instinctively, not using criteria that was scientific, but born out of a need to use organisms for our own use, for food, shelter, and clothing. Aristotle was the earliest to attempt a more scientific basis for classification. He used simple morphological characters to classify plants into trees, shrubs, and herbs. He also divided animals into two groups, those which had red blood and those that did not. In Linnaeus' time, a two kingdom system of classification with Plantae and Animalia kingdoms was developed that included all plants and animals respectively. This system did not distinguish between the eukaryotes and prokaryotes, unicellular and multicellular organisms, and photosynthetic, that is, green algae, and non photosynthetic, that is, fungi like organisms. Classification of organisms into plants and animals was easily done and was easy to understand, but a large number of organisms did not fall into either category. Hence, the two kingdom classification used for a long time was found inadequate. Besides gross morphology, a need was also felt for including other characteristics like cell structure, nature of wall, mode of nutrition, habitat, methods of reproduction, evolutionary relationships, etc. Classification systems for the living organisms have hence undergone several changes over the time. Though plant and animal kingdoms have been a constant under all different systems, the understanding of what groups or organisms be included under these kingdoms have been changing. The number and nature of other kingdoms have also been understood differently by different scientists over the time. Table 2.1 Characteristics of the five kingdoms. First is Monera. Cell type is prokaryotic. Cell wall is non-cellulosic, that is made up of polysaccharide plus amino acid. Nuclear membrane is absent. Body organization is cellular. And mode of nutrition is autotrophic, that is chemosynthetic and photosynthetic, and heterotrophic, that is saprophytic or parasitic. Next is Protista. Cell type is eukaryotic. Cell wall is present in some. Nuclear membrane is also present. Body organization is cellular. And mode of nutrition is autotrophic, that is photosynthetic and heterotrophic. Next is fungi. Cell type is eukaryotic. Cell wall is present with chitin. Nuclear membrane is present. Body organization is multicellular or loose tissue, and mode of nutrition is heterotrophic, that is saprophytic or parasitic. Next is Plantae. The cell type is eukaryotic. Cell wall is present, that is cellulosic. Nuclear membrane is also present. Body organization is tissue or organ level, and mode of nutrition is autotrophic, that is photosynthetic. Next is Animalia. Cell type is eukaryotic. Cell wall is absent. Nuclear membrane is present. Body organization is tissue or organ system level. And mode of nutrition is heterotrophic, that is holozoic or saprophytic, etc. R.H. Whittaker in 1969 proposed a five kingdom system of classification. The kingdoms defined by him were named as Monera, Protista, Fungi, Plantae, and Animalia. The main criteria for classification used by him include cell structure, body organization, mode of nutrition, reproduction, and phylogenetic relationships. The three domain system has also been proposed that divides the kingdom Monera into two domains, leaving the remaining eukaryotic kingdoms in the third domain, and thereby a six kingdom classification. You will learn about this system in detail at higher classes. Let us look at this five kingdom system of classification to understand the issues and considerations that influence the classification system. Earlier classification systems include bacteria, blue-green algae, fungi, mosses, ferns, gymnosperms, and angiosperms under plants. The character that unified this whole kingdom was that all the organisms included had cell wall in their cells. This placed together groups which widely differed in other characteristics. It brought together the prokaryotic bacteria and the blue-green algae cyanobacteria with other groups which were eukaryotic. It also grouped together the unicellular organisms and the multicellular ones. Say for example, Chlamydomonas and Spirogyra were placed together under algae. The classification did not differentiate between the heterotrophic group fungi and the autotrophic green plants, though they also showed a characteristic difference in their walls composition. The fungi had chitin in their walls, while the green plants had a cellulosic cell wall. When such characteristics were considered, the fungi were placed in a separate kingdom, kingdom fungi. All prokaryotic organisms were grouped together under kingdom Monera, and the unicellular eukaryotic organisms were placed in kingdom Protista. Kingdom Protista has also brought together Chlamydomonas, chlorella, which was earlier placed in algae within plants and both having cell walls, with Paramecium and amoeba, which were earlier placed in the animal kingdom, which lack cell wall. It has put together organisms which in earlier classifications were placed in different kingdoms. This happened because the criteria for classification changed. This kind of changes will take place in further too depending on the improvement in our understanding of characteristics and evolutionary relationships. Over the time, an attempt has been made to evolve a classification system which reflects not only the morphological, physiological, and reproductive similarities, but is also phylogenetic, that is based on evolutionary relationships. In this chapter, we will study characteristics of kingdoms Monera, Protista, and fungi of the Whittaker system of classification. The kingdoms Plantae and Animalia, commonly referred to as plant and animal kingdoms, respectively, will be dealt separately in chapters 3rd and 4th. 2.1 Kingdom Monera. Bacteria are the sole members of kingdom Monera. They are the most abundant microorganisms. Bacteria occur almost everywhere. Hundreds of bacteria are present in handful of soil. They also live in extreme habitats such as hot springs, deserts, snow, and deep oceans where very few other life forms can survive. Many of them live in or on other organisms as parasites. Bacteria are grouped under four categories based on their shape. The spherical, that is coccus, plural is cocci. The rod shaped, that is bacillus, plural is bacilli. The comma shaped, that is vibrium, plural is vibrio. And the spiral, that is spirulum, plural is spirilla. Though the bacterial structure is very simple, they are very complex in behavior. Compared to many organisms, bacteria as a group shows the most extensive metabolic diversity. Some of the bacteria are autotrophic, that is, they synthesize their own food from inorganic substrates. They may be photosynthetic autotrophic or chemosynthetic autotrophic. The vast majority of bacteria are heterotrophs, that is, they depend on other organisms or a dead organic matter for food. 2.1.1 Archaebacteria. These bacteria are special since they live in some of the most harsh habitats, such as extreme salty areas, that is, halophiles, hot springs, that is, thermoacidophiles, and marshy areas, that is, methanogens. Archaebacteria differ from other bacteria in having a different cell wall structure, and this feature is responsible for their survival in extreme conditions. Methanogens are present in the gut of several ruminant animals, such as cows and buffaloes, and they are responsible for the production of methane, that is biogas, from the dung of these animals. 2.1.1 Eubacteria. There are thousands of different eubacteria or true bacteria. They are characterized by the presence of rigid cell wall and, if motile, a flagellum. The cyanobacteria, also referred to as blue-green algae, have chlorophyll A similar to green plants and are photosynthetic autotrophs. The cyanobacteria are unicellular, colonial, or filamentous, freshwater or marine or terrestrial algae. The colonies are generally surrounded by gelatinous sheath. They often form blooms in polluted water bodies. Some of these organisms can fix atmospheric nitrogen in specialized cells called as heterocysts. Example, nostoc and anabena. Chemosynthetic autotrophic bacteria oxidize various inorganic substances such as nitrates, nitrites, and ammonia and use the released energy for the ATP production. They play a great role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. Heterotrophic bacteria are most abundant in nature. The majority are important decomposers. Many of them have a significant impact on human affairs. They are helpful in making curd from milk, production of antibiotics, fixing nitrogen in legume roots, etc. Some are pathogen causing damage to human beings, crops, farm animals, and pets. Cholera, typhoid, tetanus, citrus canker are well known diseases caused by different bacteria. Bacteria reproduce mainly by fission. Sometimes, under unfavorable conditions, they produce spores. They also reproduce by sort of sexual reproduction by adopting a primitive type of DNA transfer from one bacterium to the other. The mycoplasma are the organisms that completely lack a cell wall. They are the smallest living cells known and can survive without oxygen. Many mycoplasma are pathogenic in animals and plants. 2.2 Kingdom Protista. All single-celled eukaryotes are placed under Protista, but the boundaries of this kingdom are not well defined. What may be photosynthetic protestant to one biologist may be a plant to another. In this book, we include chrysophytes, dinoflagellates, euglenoids, slime molds, and protozoans under Protista. Members of Protista are primarily aquatic. This kingdom forms a link with the others dealing with plants, animals, and fungi. Being eukaryotes, the protestant cell body contains a well defined nucleus and other membrane bound organelles. Some have flagella or cilia. Protists reproduce asexually and sexually by a process involving cell fusion and zygote formation. 2.2.1 Chrysophytes. This group includes diatoms and golden algae, that is, desmids. They are found in fresh water as well as in marine environments. They are microscopic and float passively in water currents, that is, plankton. Most of them are photosynthetic. In diatoms, the cell walls form two thin overlapping shells which fit together as in a soap box. The walls are embedded with silica and thus the walls are indestructible. Thus, diatoms have left behind large amount of cell wall deposits in their habitat. This accumulation over billions of years is referred to as diatomaceous earth. Being gritty, this soil is used in polishing, filtration of oils and syrups. Diatoms are the chief producers in the oceans. 2.2.2 Dinoflagellates. These organisms are mostly marine and photosynthetic. They appear yellow, green, brown, blue, or red depending on the main pigments present in their cells. The cell wall has stiff cellulosic plates on their outer surface. Most of them have two flagella. One lies longitudinally and the other lies transversely in a furrow between the wall plates. Very often, red dinoflagellates, example goniolax, undergo such rapid multiplication that they make the sea appear red, that is, red tides. Toxins released by such large numbers may even kill other marine animals such as fishes. 2.2.3 Euglenoids. Majority of them are freshwater organisms found in stagnant water. Instead of cell wall, they have a protein rich layer called as pellicle, which makes their body flexible. They have two flagella, a short and a long one. Though they are photosynthetic in the presence of sunlight, when deprived of sunlight, they behave like heterotrophs by predating on other small organisms. Interestingly, the pigments of euglenoids are identical to those present in higher plants. Example, Euglena. 2.2.4 Slime molds. Slime molds are saprophytic protists. The body moves along decaying twigs and leaves engulfing organic material. Under suitable conditions, they form an aggregation called as plasmodium, which may grow and spread over several feet. During unfavorable conditions, the plasmodium differentiates and forms the fruiting bodies bearing spores at their tips. The spores possess true walls. They are extremely resistant and survive for many years, even under adverse conditions. The spores are dispersed by air currents. 2.2.5 Protozoans. All protozoans are heterotrophs and live as predators or parasites. They are believed to be primitive relatives of animals. There are four major groups of protozoans. Amoeboid protozoans, that is, these organisms live in freshwater, seawater, or moist soil. They move and capture their prey by putting out pseudopodia, that is, false feet as in amoeba. Marine forms have silica shells on their surface. Some of them, such as entamoeba, are parasites. Next is flagellated protozoans. The members of this group are either free living or parasitic. They have flagella. The parasitic form causes diseases such as sleeping sickness, example, trypanosoma. Next is ciliated protozoans. These are aquatic, actively moving organisms. Because of the presence of thousands of cilia, they have a cavity, that is gullet, that opens to the outside of the cell surface. The coordinated movement of rows of the cilia causes the water laden with food to be steered into the gullet. Example, paramecium. Next is sporozoans. This includes diverse organisms that have an infectious spore like stage in their life cycle. The most notorious is Plasmodium, that is malarial parasite, which causes malaria, a disease which has a staggering effect on human population.